I'm Daniel Levine, and this is the Bio Report. Antibody-drug conjugates can deliver chemotherapy directly to tumors, but more than 95% of the dose often ends up in healthy tissues, decreasing efficacy and increasing toxicity. Mythic Therapeutics FateControl technology improves the uptake of ADCs in cancerous cells while avoiding the release of their toxic payloads in healthy ones. This promises to increase efficacy of these therapies in a wide range of cancers without causing unacceptable side effects. We spoke to Brian Fiske, co-founder and chief scientific officer of Mythic, about the challenges that have held back the benefits of ADCs, how the company's FateControl technology addresses those, and how it's thinking about building a pipeline of ADCs across indications. Brian, thanks for joining us.
Thanks for having me.
We're going to talk about antibody-drug conjugates, Mythic Therapeutics, and its FateControl technology designed to increase the uptake of ADC in cancer cells while avoiding healthy ones. Let's start with antibody-drug conjugates. For listeners not familiar with these therapies, can you explain what they are and how they work?
Absolutely. So, the original idea of an antibody-drug conjugate was to take chemotherapy, which is effective, but as we all know, can be very toxic, and improve its selectivity for tumor cells and improve its potency for tumor cells. And the way that this was originally accomplished and still is today is by attaching that chemotherapy to an antibody. And so there are several components of an ADC. The antibody component binds to a target that's found on tumor cells, can also be found on normal cells, but more selective for cancer cells. The drug portion of the ADC is typically a chemotherapy drug. And then the conjugation, oftentimes called the linker, is what attaches the chemotherapy to the antibody. And so if you dose an ADC into a patient, the idea is that the antibody component will bind the target on tumor cells. That'll cause the whole complex to become internalized into those cancer cells. And then the linker is then designed to release that chemotherapeutic payload, which then goes on to kill the cancer cells. And so ultimately, our best data to date would show that that's resulted in about a 10-fold increase in selectivity for delivering chemotherapeutic payloads to cancer cells. But even our best ADCs today are only able to deliver about 1% of any given dose to tumors. So it still potentially has a long way to go in terms of being truly selective for cancer cells.
It's now been almost 25 years since the first ADC won approval. There are now about 15 on the market. What's been the experience with these? And you say they've only been able to take up about 1%. Why is that? Why are they not able to more effectively deliver these therapies?
Sure. So maybe I'll address the second part of the question first. Our best data would really come from two sources. One is looking at dosing ADCs into relevant animal models, where we have an animal with a tumor and we can dose the ADC to treat that tumor. And that data would say you get about a 10-fold concentration of the ADC into tumors versus other healthy tissue. But a typical patient who's having a really hard time with their cancer might be less than 1% by mass tumor. And so even with a 10-fold concentration, you may only end up at 1 or a couple of percent. And that's confirmed by imaging studies where you can label antibodies or ADCs with imaging agents and then do scans to find out where the ADC distributes. I think the good news is, even with that surprisingly low level of selectivity, but with that increase in selectivity, when ADCs work, they work really well. So if you are a cancer patient who learns that they can be treated with an ADC, that's really great news. And at Mythic, we just think it doesn't happen often enough because due to that, long ways to go maybe in terms of selectivity, currently ADCs are really restricted to small subsets of patients or small subsets of targets. And those targets and patients that really respond well are those with very high levels of expression, right? So hundreds of thousands of copies per cell, which is quite a lot. And so that's the problem we're interested in solving at Mythic. And we think if we could solve it, it would really unlock the full potential of ADCs, which is hopefully in the future to replace chemotherapy.
And to what extent have the existing therapies been able to address the challenge of toxicity?
So I think partially, right? They are an improvement over chemotherapy, as we've been discussing, but I think they have potentially a long way to go. ADCs are typically, like many other chemotherapy drugs, dosed to so-called dose-limiting toxicities. And so the dose that we pick is generally at or close to the highest dose we can achieve in consideration of those toxicities, because we want to maximize efficacy, because the types of cancers we're going after in general are deadly if we can't shrink them. And so ultimately it depends on the dose and the patient, but ADCs might have less toxicity than something like chemotherapy, but I think we could do better.
My sense has been that the challenge with ADCs and the magic of them has always been around the linker. Is that the case?
Yeah, so I think now, and if you look historically at the field, the focus of all the major players, of all the startups, is really on the linker payload piece. And that's because there are a number of really hard challenges to solve with linker payloads. Obviously, we have a hypothesis at Mythic that actually the huge renaissance, if you will, in ADCs over the last few years, going from 2 products in 2017 when we started in ADCs to now, as I think you pointed out, around 15 approved therapies, is really due to the field finally understanding and unlocking the linker toxin piece. But we see the road ahead as much more focused on the antibody component.
What other challenges need to be addressed to really expand the field of ADCs?
Yeah, so I think maybe to touch on historically, there have been a number of other challenges besides the linker to be addressed. So things like manufacturing. So antibodies in general are a very nicely behaved manufacturable modality, but when you put linker toxin on them, they don't produce as well. And so manufacturing can be challenging. I think it's also been challenging, and as a field, we've had to learn through clinical data to understand how to match the potency and mechanism of action of different payloads to different indications. I think the current challenge we're facing is really around these limited patient populations. But another challenge I would put out there, which maybe we can contribute to at Mythic, but I think is a big enough challenge where lots of other folks can dig in, is expanding the use of ADCs beyond cytotoxics for oncology. So the field is very focused on payloads and drugs that kill cells and then directing those to tumors. But what if we could deliver siRNAs? What if we could deliver steroids? What if we could use those different payloads to treat other diseases besides cancer? I think that's also an exciting next challenge to take on as a field.
Mythic has developed what it calls its FateControl technology. What is this and how does it work?
Sure. So we use FateControl to mean the idea or the concept of delivering more payload where you want it in tumor cells and less where you don't in normal cells. And our focus for this technology, as compared to where the field has been at historically, as we've been discussing on linker payloads, is on the antibody component. And so we have a first generation technology based on pH engineering, and I can get into a little bit of how that works, but we're also developing next generations of technology to address the same problem. How do we take ADCs from being 1% specific in delivering 1% of their dose to tumor cells to, closer to the full potential of 10, 20, 100%.
How does this change what a Mythic antibody-drug conjugate consists of? How does it alter the components of an ADC, or would it appear like any other ADC to someone?
Yeah, I think the really exciting thing that we've been able to accomplish is actually not changing the format of an ADC. I think, we're very proud and feel fortunate to stand on the shoulders of giants in the sense that the field has progressed a ton over the last 40 years. And most of that progress has come from conjugating a select class of payloads, really tubulin toxins and topoisomerase inhibitors, to IgG-like molecules using a peptide-based linker. I mean, that's a little bit technical, but people have found sort of favored solutions to classic problems with antibody format and linker and payload. And we don't really want to change anything there that's working. We want to focus on our very specific problem and ultimately have the same format as other ADCs.
Mythic's lead experimental therapy is MYTX-011. This is an ADC in development for non-small cell lung cancer. Given the potentially wide applications for this technology, why start with that indication?
A great question. So we're starting there. I don't think we'll stop there. That's maybe the first thing to say. No one is at Mythic because we want to become a single product company. We want to become a enduring multi-product oncology company. Non-small cell lung cancer is a very large population, and that very large population has very high unmet need. It's a very deadly cancer, causes a lot of problems. Patients progress on existing therapies. And so from an unmet need and market perspective and patient impact perspective, it's certainly an interesting one. I think the other thing about the indication is that for MET ADCs specifically, there was already clinical data to say that essentially more is better, that if we could deliver more payload, that we would have better responses in patients. And that's because existing MET ADCs have shown that they can be a really great drug for the highest expressors with the most sensitive histotype and genotype of cancer. So those 5 to 10% of patients who express the highest levels of MET can respond really well to existing ADCs, but we know that there's another 20 to 25% of patients with lower levels of expression, so-called intermediate levels of expression. We know there's another 40% of patients beyond that even who have lower levels of expression. And so it's really exciting to think about that existing proof of concept that those patients with the highest levels are responding, that patients with even intermediate levels are not, and that the barrier therefore is really how much toxin can you deliver. And maybe what we can do at Mythic is make the ADC do that work and that heavy lifting rather than having the patient do it themselves by happening to have highest expression on their tumors.
What are the components of your lead experimental therapy, and how does it work?
The specifics, so we have a pH engineered c-MET antibody that's actually based on a scaffold that's already been in clinic before, not with us, but with another company. So we've pH engineered it to bind at neutral pH, to bind at the slightly acidic pH of the so-called tumor microenvironment, but to specifically release from the target when it gets inside of cells. And that antibody is then attached to a very standard linker toxin, VC-MMAE, that's actually been tried with MET ADCs before and been successful. And so our hypothesis is really around that the pH-sensitive antibody can deliver more, and that's what we found in preclinical data, and that's what we're trying to prove out now with clinical data.
What's the development path forward?
So we're starting with monotherapy in non-small cell lung cancer, where we feel like there's the most data and most existing precedent. But I think once we prove that out, we'll be looking at combinations. We'll be looking at other indications that express MET. And some of those indications are really exciting because we see indications where MET is expressed broadly, but those indications lack that highest expressing population that's being treated now in non-small cell lung cancer. And so looking at indications where MET expression is very widespread, but you have really just intermediate and low expressors is super exciting for us because that's exactly where we've designed our drug to be differentiated.
And how are you determining other indications you'll pursue, and what's the strategy for building a pipeline?
We're pretty indication agnostic from a technology perspective. We've gone very broad with our platform and our pipeline. We've worked with over 20 different targets in our lab. We've put our engineering into over 100 different clinical stage ADC antibodies. We're on candidate number 15,000 or something like that at this point. So we're going very broad. I think we'd look for some of the same things I talked about with non-small cell lung cancer. Where do you have big populations of patients who aren't well served with existing ADCs? And at least initially going after targets where it's already pretty clear from existing clinical data that more is better, that if you could deliver more, you'd have a better outcome.
And are you looking to leverage the platform through partnerships?
Absolutely. So we're a small company, but we've seen big things from what our platform can do, and we won't be able to pursue all of those things to have a real impact on patients. And so, we are looking for partnerships. Our ideal partners would bring capabilities to the table that would help expand the impact of what we do. So those could be capabilities with new payloads. I mean, we can use a lot of different payloads at Mythic, but one way of understanding Mythic is we're the only ADC company that's not working on linker payloads. So we're not working on anything novel on that side. And so if a partner would bring a capability of new payloads, that could be really interesting. We could look at partners who bring expertise in certain indications, and some of those indications could be outside of oncology. So looking at delivering novel payloads for autoimmune, siRNAs, et cetera, et cetera.
You mentioned that there are other companies out there that are now seeking to develop other conjugated therapies. How translatable is your approach to doing something like an oligonucleotide that's conjugated to an antibody?
I'll make clear, I guess, what we've done and what we haven't done. We haven't tried conjugating an oligonucleotide or a steroid or really anything outside of a cytotoxic. But what we've seen with our internal focus on oncology is that we are truly payload agnostic, right? Every type of payload that we've tried, we've been able to deliver more. And it kind of makes sense because, if you were to take an antibody-drug conjugate and blow it up to the size where you could actually see it, the ADC is like 99% antibody by volume. The antibody is huge, the toxin is fairly tiny. And so the properties, it's not only that the antibody component its job is to deliver it within the body and define where it goes, but also just by mass, it's the biggest piece. So we've seen that our platform across several classes of toxin, tubulin toxins, DNA toxins, topoisomerase toxins, again, all focused on oncology internally, we're completely agnostic and we can deliver more and we can see greater efficacy. So I think one space that we're probably, at least for now, the next couple of years, we're probably not going to get to, but would be really exciting, is delivering other types of payloads. And my understanding of some of the challenges with these other payloads, and frankly, the reason why ADCs have been a lot of impact on oncology so far as compared to other fields, is that these other payloads suffer from issues due to low potency, and that's exactly the problem we solve. So that could be quite exciting.
The other thing that interests me about this is that the antibodies are often viewed merely as a targeting mechanism, but do you ever look for antibodies that could have a therapeutic benefit as well?
It's a great question. So, stepping back for a moment, I think some of the most effective drugs have multiple mechanisms of action. So, for example, Herceptin, right? Herceptin is everyone's favorite antibody against HER2, which is a target in breast cancer. Herceptin blocks HER2 signaling. HER2 can act as an oncogene in certain tumors and drive tumor growth, so you can block that with the antibody. But you also end up, by decorating the surface with antibodies, you also end up having immune cells come in, recognize those antibodies, and start attacking the cancer cells. And then on top of that, one of the biggest stories in ADCs in the last couple of years has been in HER2, where on top of those already two mechanisms of action, at least, they've added an ADC mechanism of action. So that's been a really exciting story to watch. That's an example of a drug that has multiple MOAs. And I think MYTX11 has that same sort of potential, especially with regards to the fact that MET is a known oncogene for certain subsets of cancers. I think we're looking to hit a broader population of patients who are not strictly dependent on MET signaling, but could blocking MET via our antibody have a partial effect that would synergize with the ADC mechanism of action? That's very possible, and it's quite exciting to think about.
The company completed $103 million Series B financing at the end of 2020. How far will existing cash take you, and what's the plan for raising additional capital?
Sure. So we are funded through end of this year, 2024, and that will take us through readout on our dose escalation portion of our trial, and also we'll be reading out a number of programs in our pipeline to what we call the development candidate stage, and that's the stage where we can start IND-enabling work. So we are excited to go out this year and fundraise. We're fairly early in our thinking in terms of when and how much and who, et cetera. But I would expect I would expect we're going out as early as Q2. And yeah, we'll be excited to be out in fundraising. It seems like a more favorable market than at least the last few years.
Brian Fiske, co-founder and chief scientific officer of Mythic Therapeutics. Brian, thanks so much for your time today.
Thanks, Danny.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.